Hello students, in last video we have studied mitosis. So in this video we are going to study what type of cell division, and that type of cell division is their meiosis. So in case of the meiosis, already we have studied what is meant by mitosis. Mitosis is a type of cell division and in which chromosomal number remains diploid. That means in the multiple of 2. So chromosomal number is remaining diploid in the mitosis, but in the meiosis that chromosomal number get reduced to half. That chromosomal number is getting reduced to half. Suppose initially 2n number is there in the initial before the meiosis. Suppose that cell number in the cell chromosomes are there 2n, but in the meiosis, at the end of the meiosis, chromosomal number becomes only n. That means it gets reduced to half. So we are going to learn about meiosis, and meiosis is important concept, and in that meiosis you should know different concept before understanding that particular meiosis. And that concept, 2-3 concepts are there, and if you will understand that 2-3 concept, you can understand that meiosis. So first important thing is there chromosomes. You are knowing chromosomes, and in that chromosome we have studied chromosomes. Upper arms are there, lower arms are there, P arm, Q arm, you are saying, and at the center centromere is there, at the middle centromere is there. So suppose chromosome is like this. Suppose chromosome is like this. So this is chromosome, and this one is there P arm, this one is there Q arm. So these two, this P arm, this Q arm, this upper part and this lower part, these you are saying sister chromatids. Sister chromatids. So these two are the parts of same chromosome. These are the two arms or two parts of the same chromosome. So that's why these are called as the sister chromatids. And if similarly another same chromosome is there, another chromosome is there, and this one, these are chromatids. These are also sister chromatids, but suppose this P arm, this one is the Q arm. These two, P arm, Q arm, these are two sister chromatids of this. Suppose this one is chromosome number 1, this one is chromosome number 2. So in this first chromosome, these two are the sister chromatids, and in the second chromosome these two are the sister chromatids. But this uppermost part and this part, you cannot say these are two sister chromatids, because these are the parts of different chromosomes. These are the parts of different chromosomes. So this part and this part you can say non-sister chromatids. These two, they separate parts. These are called as a non-sister chromatids. Non-sister chromatids. So sister chromatids and non-sister chromatids. Sister chromatid, when you are saying, when these two parts are there of the chromatids, these are of the same chromosome, then sister. And if these are of sister chromatids of different chromosome, then you are saying these are the non-sister chromatids. So if you will understand this one, then it is easy. The next thing is there, another confusion may be there, centromere and centriole. That another confusion is there, centromere, and another one is there, centriole. I think this confusion will be there for you. So suppose this one is cell, this one is there cell membrane, and in this one is there nuclear membrane, Again, in the nucleus there are chromosomes. There are chromosomes. Like this way chromosomes are there. So, and in this one circular nucleolus is there. What is there? Nucleolus. So, and outside the nucleus, here this one is there. You can say centriole. This one is there, centriole, and at the middle of the chromosome, where two arms are joined, at the center of the chromosome, this one is there, centromere. So these 2 points should be clear: centriole and a centromere. Centriole is there in the cell, but outside the nucleus, and centromere is there at the middle of the chromosome. This one is also important. Now we are going to study about meiosis, and that meiosis we are going to study in detail. So in case of the meiosis, first thing is there, it occurs in two stages. Meiosis first and meiosis second. In that, meiosis first and meiosis second is there. So two stages are there in the meiosis. In that, first one is there, meiosis first, and second one is there, meiosis second. So in this meiosis first, second thing is there, meiosis first is, you can say it is lengthy. It is a long process. Lengthy or long process, because it takes more time, and in these stages are also different. So in the long process, in that meiosis first, whatever prophase is there, that prophase is having 5 stages. In the meiosis first, prophase is having 5 stages. So 5 steps are there. In that, first one is there, in the prophase, first one is there, what you can say, prophase first. In that, 5 stages are there, and first one is there, leptotene. Second one is there, zygotine. Third one is there, pachytene. Fourth one is there, diplotene. Diplotene. And fifth one is there, dikinesis. So in the meiosis first, prophase is there, and that prophase first is having 5 stages or 5 steps are there. Which steps are there? leptotene is there, zygotene is there, pachytene is there, diplotene is there, and dikinesis. So these 5 steps we are going to learn in detail. So why you have to learn this one? Because in this reasons are there. First thing is there, in the prophase first, crossing over takes place. So crossing over between non-sister chromatid takes place. So before this I explain what is the non-sister chromatid. That means chromatids of different chromosomes, crossing over between the chromatids of different chromosomes. That means one chromosome, it's one sister chromatid, and another chromosome and it's another sister chromatid, and between these crossing over takes place, exchange of genetic material takes place, and because of that crossing over takes place, and because of that, second thing is the genetic variation takes place. Genetic variation, that means whatever gametes are produced, or germ cells are divided, these are having different genetic recombination. Genetic combination is different. So we are going to learn meiosis in detail. And important thing is there, meiosis occurs in the germ cells. In which cells meiosis occurs? Meiosis occurs into the germ cells. This meiosis occurs in the germ cells. Germ cell, you can say reproductive cells. Reproductive cells. So important thing is there, meiosis occurs in the germ cells, mitosis occurs into the somatic cells or stem cells. So many times we'll repeat this one, because it is, you have to listen it, you have to look this video carefully, then only you can understand this one. Because these concepts before this or now also these are there in the 11th and 12th standard. So from first we are going to learn this one, just give attention. So listen, see, in case of the meiosis, already these things I explained. Now we are going to learn prophase, meiosis first, and in that all stages we are going to learn. So meiosis we are learning, and meiosis occurs in 2 stages and that stages we are going to learn. So in case of the meiosis, first we are going to learn, first stage we are going to learn, and that one is there, meiosis first. Meiosis first. So in this, different things are there, that's why I am dividing this board. Meiosis first. In that, first stage is there. First stage is there, liptoting, or we'll take stage, or you can say step. First step is there, step first, prophase first. And in that prophase first, already we have studied what do you mean by prophase. What do you mean by prophase? Then in the meiosis, mitosis we have studied prophase, metaphase, anaphase, telophase. These phases you are knowing. So in case of the prophase first, so what happens in the prophase first? There are again 5 steps are there. In the prophase first, there are 5 steps, or you can say stages are there. Suppose in this 5 stages are there. Which 5 stages are there? That one is there. First, liptoting. So what occurs into the liptoting? We'll study with the help of diagram. In case of the diagram, and with the help of diagram it is easy to understand. So in case of the liptoting, we'll draw one diagram. This one is the cell. In that cell membrane is there, and inside nucleus is there. Inside nucleus is there. Suppose, and in that nucleus, suppose this one is there, nuclear membrane. This one is there, nuclear membrane. This one is there, cell membrane. Okay. Another thing, in this we are going to show 4 chromosomes, then it will be easy to understand. So suppose 4 chromosomes you will take. Suppose one chromosome is like this, or you can draw like this. Suppose second one is like this, or for understanding we'll take, suppose one chromosome is like this, another chromosome is like this, or third chromosome is like this, and fourth one will be like this. So in the prophase first, prophase first in that liptotin, which things occur? Which things occur? And in this, I told before, here will be centriole. Here will be centriole, from where spindle fibers develop. From where spindle fiber develop. And here will be nucleolus. And this one is their nuclear membrane. This one is their nuclear membrane. So what takes place? Actually, what takes place in this? So in case of the liptotin, condensation of chromosomes takes place. Condensation of chromosomes takes place, and that chromosomes are becoming, second thing, chromosomes become compact and thick. Chromosomes become compact and thick. So this occurs in case of the meiosis first, in that prophase first, and first stage is the liptotin. So second stage is there in the prophase first, that one is their zygotin. That one is their zygotin. So in the zygotin, which changes takes place, that one we see. So in case of the zygote, firstly we will draw diagram. This one is the nuclear membrane. Here centrioles, they will get divided. Centriole get divided here, and after this centriole start to divide. Again important thing is there. Again important thing in case of the centriole, or in case here we are drawing nuclear membrane. So in the zygotin, whatever 4 chromosomes are there, their pairing starts. So pairing in case of the homologous chromosome takes place, and how that pairing takes place, that one we see. Suppose here one chromosome is there, like this way one chromosome is there. So another chromosome, it will start pairing here. So pairing of that particular chromosomes takes place. Pairing of that particular chromosomes takes place. So like this way, these chromosomes will come close to each other. These chromosomes will come close to each other. And suppose here nucleolus is there. Nucleolus. So here nucleolus is there, and in the zygotin these chromosomes are coming close to each other. So pairing of chromosomes, and these are called as paired chromosomes. Paired chromosomes. Okay, pairing of the chromosome takes place, and a synaptical complex is produced. Synaptomial. So here nearby pairing takes place, and here whatever there, and it is called as a synaptomial. Or you can say synaptomial complex. What you are saying? Synaptomial complex. So after this, and this one you are saying synapsis takes place. So in the liptotin, condensation will start. In the zygotin, these chromosomes start to pair. So explanation in that, pairing of chromosome takes place, or you can say homologous chromosomes. start to pair. Homologous chromosomes start to pair, and synaptomial complex is formed. Synaptomial complex is formed. So third stage we'll study, and that third stage is there pachytene. So this one is important step. Pachytene is the important step. Diagrammatically we'll see, in case of the pachytene, suppose this one is the nuclear membrane, here this one is the cell membrane, this one will be nuclear membrane. Nuclear membrane is there, cell membrane and nuclear membrane. So again here whatever centriole is there, that centrioles get separated. One centriole move here, one centriole will move towards the downward pole, and one will move to the upward, and here spindle fibers will develop. But in case of the, suppose we will show only one, like this way spindle fibers will develop. So in the pachytene, crossing over between the chromosomes takes place. And in that pachytene, we'll see what takes place actually. So in the pachytene, suppose you will consider one chromosome is here, its one arm is here, another arm here, another chromosome. So in this, suppose another chromosome, its one arm will cross over here, another arm will be there, one arm will cross over here, and one arm will be like this, and like this way second two chromosomes, they will be like this. So one arm is here, another arm is here, one arm is here, another arm is there. And in case of next chromosome, one arm will be here, another arm will be here, next arm like this way. So crossing over between the chromosomes takes place. In case of the zygotine, in case of the pachytene. So what takes place in the pachytene, that one will see. Or crossing over, we can show in a little bit more complex. So for understanding only, we have shown one arm only crossover, like this way crossing. But actually crossing over you can show like this way crossing over takes place. But for understanding we are showing just like this crossing over. So in that crossing over takes place. So names will give to this diagram, and that names are there. First one is there crossing over chromosomes. You can give name here, crossed over chromosomes. Again here will be nucleolus. Again here centromeres are there. Centrioles are there. Centrioles get separated. So which changes takes place in this stage we are going to learn. So first change is there. So crossing over between non-sister chromatid takes place. What you are saying? Crossing over between non-sister chromatids takes place. So this one chromosome is sister chromatid, this one chromosome is sister chromatid, but these sister chromatids get crossed over, but these are not similar of the same chromosome. One sister chromatid of one chromosome, another sister chromatid of another chromosome, and crossing over between non-sister chromatid takes place. Second thing is the genetic recombination takes place. Genetic recombination takes place. Genetic recombination takes place. Third thing is there, homologous chromosomes, they remain paired at the site of the crossing over. So homologous chromosomes remain paired at site of crossing over, and they are forming crossing over, and so these three changes we have studied. Now fourth step is there, fourth stage is there, and that fourth stage is there, diploting. Fourth stage in the meiosis, first in that prophase first. So in the diploting, which changes takes place, that one will see. So in case of the diploting, this one will be cell membrane, this one is there, cell membrane, then here will be nuclear membrane, here will be nuclear membrane is there. Another thing, centriole, you can show one centriole here, another centriole will be here. So centrioles will reach to the opposite poles, and a spindle fiber is there between them. So another thing, in case of the diploting, now these crossover chromosomes will get separated. In the diploting, crossover chromosomes will get separated, and they will remain in the crossover, but where? At the site of pairing only, at the site of pairing. So you can show like this. One is here, another one like this way, but they will get separated totally, and they would remain crossover at the site of here, at the site of crossing over, they will remain in touch. Another thing is that here one chromosome is like this, another chromosome is here, so they will get separated, and one will be like this, another like this, and here we have shown these are very close to each other. Here we have shown these are very close. Actually these are like this, actually like these are like that, rolled or coiled, but we are shown like this, close to each other, and here they are little bit away from each other, little bit away from each other. So what you can say, what you can give name here, here chiasma, you can say chiasma. Chiasma means like that multiple sign is there, or cross mark, like this way mark is formed here between non-sister chromatids, and so you are saying this one is a chiasma. Or in case of the optic chiasma, like this way chiasma is there. And which changes takes place in this diploting, we'll see. In this, first thing is there, so synaptomial complex dissolves and homologous chromosomes get separated except point of crossing over. Homo, or what you can say, synaptomial complex, synaptomial complex get dissolved, and homologous chromosomes get separated, separated except what you can say, except at the point of crossing over, at the point of crossing over, at the point of crossing over. Second thing is there, and crossing over looks like except, and it is called as a chiasma or chiasma. Crossing over looks like except, called chiasma. It is called as a chiasma. So these are the 4 stages. Now in the 5th stage, here less is less, but I will show. So in case of the 5th stage, and 5th stage is the dikinesis. 5th stage is there in the prophase first, and that one is their dikinesis. And which changes takes place here? So I think you have understood this one. I will show here 5th stage. Here less is less. So at the first stage I will show 5th stage. So one more time we'll see what happens here. In the prophase first, 5 stages are there, and first one is the liptotin. In the liptotin, condensation of chromosome takes place and chromosomes become compact and thick. In the centriole is there, nucleolus is there, cell membrane is there, nuclear membrane is there. Again, zygote is there, and these chromosomes, they are coming near to each other. They will get paired. So they will get paired here, paired chromosomes. Then cell membrane is there, nuclear membrane will be here, nucleolus is there, and centriole is there. This one centriole is there. This one centriole. Okay. So homologous chromosomes start to pair. Synaptomial complex is formed, and in the 3rd step is the pachitin. In the pachitin, crossing over of that particular chromosome, non-sister chromatid takes place. Crossing over between non-sister chromatid takes place. Cell membrane, nuclear membrane, crossover chromosomes, nucleolus is there, and centriole is there. And like this way, they would cross to each other. Non-sister chromatid. Suppose one chromosome here, and another centromere is here. So another centromere will be here. So like this way, they will coil, they will cross over in the pachitin. So crossing over between non-sister chromatid takes place, genetic recombination takes place, and homologous chromosomes, they remain paired at the site of the crossing over. And in this crossing over, whatever genetic materials or that particular non-sister chromatids, in these non-sister chromatids, genetic material is exchanged. It is exchanged. And 4th stage is there, diplotin. In the diplotin, these crossover chromosomes, they will get separated. Cell membrane is there, nuclear membrane is there. Again, another thing is there, centriole is there. Centrioles, one will move upper part, one will move to the lower part, and synaptomial complex, where crossover, that complex will get separated. Chromosomes will get separated, but they will remain crossed at the site of the crossing over only, at the site of pairing. Synaptomial complex get dissolved and homologous chromosomes get separated, except at the point of the crossing over. And crossing over looks like except, and it is called as a chiasma. And the 5th step I am going to show here. Step number 5th, or stage number 5th. In that, what takes place, we'll see. Stage number 5, in case of the prophase of meiosis first. What we are learning? Prophase first. In that 5 stages, 5th stage is there dikinesis. 5th stage is there dikinesis. So in this dikinesis, what takes place? That one will see diagrammatically. Cell membrane is there, or you can cell membrane. Now here, so whatever nuclear membrane is there, it will start to disappear. Again, in this, suppose 4 chromosomes we have shown there. These chromosomes will get separated, and that crossing over will also get separated. Suppose 2 chromosomes, like this. So here one is there, here another. I can see here one is there, here another. So 4 chromosomes are there. And in this, whatever centrioles are there, they are moving to the opposite poles. They will get separated, they will move to the opposite poles. And whatever nuclear or nucleolus was there, it was completely dissolved or completely get disappeared. So what you can say here, spindle fibers are produced. Spindle fibers are produced. So this one is the prophase first, and that chromosomes are becoming thick and condensed. Chromosomes, thick and condensed chromosomes, and it will be like that of the prophase of the meiosis or prophase of the mitosis. Okay, so in the prophase, what takes place, that one we have studied. In case of the mitosis, in the mitosis we have studied prophase. In that same stages are there. In the prophase, nuclear membrane will disappear. Okay, we'll take what are the points in this. So first important thing is there, termination of chiasmata will take place here. In this termination of chiasma, that means whatever X is there, that one is removed. Now it will end of the chiasma. That means chromosomes get completely separated. Chromosomes get, here we are not showing crossing over, they are shown separately. So in the 5th stage, chromosomes get separated completely. Another thing is there, nucleolus. Second thing is there, nucleolus. It will disappear. Disappears. Third thing is there, nuclear membrane also start to disappear. Nuclear membrane also starts to disappear. So all these stages we have studied in the prophase first of the meiosis first. Now prophase first, in that 5th stage, then metaphase first, anaphase first, telophase first, we are going to study in continuous. So these stages you have studied. Now after this, we'll see what takes place. So this one is concept easy for understanding, but you have to pay attention properly. So prophase first, and in that we have studied last stage is there. Which one? Last stage we have studied. Suppose their membrane, it start to disappear. So last stage is the dikinesis. In that we have studied here one chromosome is there, here another chromosome, suppose here next chromosome, and like this way chromosomes are there. Prophase first, and this one will be dikinesis. Last step is there. So after this, what takes place? This one is the prophase first. So we are going to study this one is there metaphase first. In the metaphase first. So in the metaphase, what takes place? Suppose we will show this cell like this. We will show this cell like this. So this one will be cell membrane. Then after that we have seen here will be okay. So here will be centrioles. This one is their centriole. So in the metaphase first, what takes place? So suppose chromosomes, like this chromosomes are there. Another chromosome will be here. Another thing, these chromosomes will show it a different color. So in the metaphase we have studied these chromosomes are brought to the equatorial plane. They are coming to the equatorial plane. Equatorial plane. They are coming to the equatorial plane. Here will be spindle fibers. Spindle fibers. So we have studied. So spindle fibers will get developed between. This one will be here. It will be centromere. Centromere, and this one is centriole. So spindle fibers are developed between centriole and centromere. Here also centriole and centriole. Centriole and centromere. Spindle fibers are developed between centriole and centromere. So because of that, that chromosomes are brought to the equatorial plane. So next stage is there. Already these stages we have studied in the mitosis. So next stage is there metaphase, then anaphase. Anaphase first. So what will take place here? So like this way we are going to show. So in this, suppose these non-sister chromatids. Suppose these are pooled. Here centriole is there. Here also centriole. So these chromosomes are pulled towards the opposite poles. Centriole is there. In this anaphase, what takes place, which changes takes place, I will tell orally only. So in the anaphase, whatever centromere is there, it will split, and that sister chromatids or sister chromatids, these are pulled towards the opposite pole. So two chromosomes are pulled in upward direction, two are pulled in downward direction. After this, next step is there. Whatever next step is there, that one is the telophase. Telophase. First, in the telophase, first, suppose this one metaphase, telophase first. So in the telophase, we have studied, actually centrally notch is produced in case of the animal cell, and in case of the plant cell plate is produced, and because of that two cells get separated. Now here nuclear membrane will reappear, here also nuclear membrane will reappear, then here centromere will be there, centriole will be there, centriole, here also it will be centriole. Again here suppose two chromosomes are there, one chromosome will be like this and another chromosome will be like this. So, and here also one chromosome like this and another chromosome like this. So, and here nucleolus will reproduce, nuclear membrane will develop, nuclear membrane is produced. Here also same thing is there, nucleolus, this one will be nuclear membrane, it will be nuclear membrane, and this one will be cell membrane, this one will be cell membrane. So what takes place in this, we have studied. So this one is the prophase first, in that last one is the dikinesis, 4 chromosomes are there. In the metaphase, first these chromosomes are brought to the equatorial plane, the nuclear membrane will completely disappear. Again in the anaphase, these chromosomes, they will split at the centromere and they will get pulled to the upper pole and lower pole. And in the last phase we have studied, in case of the telophase first, two nucleus are produced, and in the cytokinesis, that cytoplasm will get divided and the two daughter cells are produced. What is produced? Two daughter cells, or you can say two daughter cells or haploid cells are produced. Okay, suppose we will say daughter cells are produced, and these are haploid, means here 4 chromosomes are there and here 2 are there. That means 4n is there, it is reduced to 2n, it is reduced to 2n. That means 4n and number is divided and 2 cells are produced. One is cell here and another one is here. So this one is there, meiosis first. This one is there, meiosis first. And this meiosis second is similar to the mitosis. Meiosis second is similar to the mitosis, and mitosis in that we have studied which stages are there: prophase, metaphase, anaphase, telophase, and after that cytokinesis is there. So you have, I think you have understood from the 4 chromosomal cell is having 4 chromosomes, but after that metaphase, anaphase, telophase, two cells are produced, and that two cells are having half number of chromosomes. Here 4 and here 2 twos are there. 2 chromosome, 2 chromosome, 2 cells are produced. 2 cells are produced. Up to this, next part is there. Next part is there, meiosis second. Next part is there, meiosis second. So meiosis is easy. If you will understand mitosis, then meiosis is so much easy. So next thing, meiosis second. We are learning meiosis. In that meiosis first over, now we are learning meiosis. Meiosis second. So meiosis second is similar to mitosis. That means which stages are there in the mitosis? Prophase second, in that metaphase second, anaphase second, and telophase second. Only, and these phases are there in the mitosis also. Mitosis. So meiosis second is similar to the mitosis because 4 stages or 4 phases are there. So in this already we have studied in the meiosis first, 2 daughter cells are produced, and these 2 daughter cells, these are having chromosomes. How many chromosomes are there? 2 chromosomes are there. Suppose one is here like this, and another one is here like this. So one is here, another one like this. 2 chromosomes. And suppose this one is there. Prophase second. In the prophase second, what will take place? Nuclear membrane will start to disappear. Whatever nucleolus, it will also start to disappear. Here also nucleolus is there, it will also start to disappear. Nuclear membrane will start to disappear. Chromosomes are becoming thick and short. Chromosomes are becoming thick and short. They will become thick and they will become short. Thick and short. So up to this, next phase is there, metaphase. Metaphase second. Which phase is there? Metaphase second. In the metaphase second, whatever these chromosomes are there, so here centriole will get separated. Centrioles. Centriole. Here also centriole will move to the opposite pole. Here also centriole move to the opposite pole. Here also centriole to the opposite poles. And these chromosomes, these are brought. These chromosomes are brought to the equatorial plane. These chromosomes are brought to the equatorial plane. Chromosomes brought to the equator, and spindle fibers will get developed. Spindle fibers are produced between, this one is their centriole, and here will be centromere. Spindle fibers get developed between centriole and centromere. Here also same, centriole, and here will be centromere, and here will be equatorial plane. equatorial plane. So after this, next stage is there, and next stage is there, that one is there, anaphase. Suppose next one is there, anaphase. Anaphase second. In case of anaphase second, one cell is here. In the anaphase second, centriole is here, centriole is here, centriole here, centriole is here. So chromosome will split. Chromosome will split. And these chromosomes after splitting, they will be pulled towards the centriole. They will pull towards the centriole. They will get pulled towards the centriole in the anaphase. So in the last phase, here also you have to show spindle fibers. So here centriole and a sister chromosome. Here you can say sister chromosome. So, and the last page is there now. Last page. Which one is there? So, suppose upper cell is there, and this one is there. Lower cell is there, and here upper cell, lower cell. Now here in this, what you can say, after this centrally notch will be produced, notch will be produced, and here nuclear membrane will be produced, and here also nuclear membrane, here also nuclear membrane, and it will get separated from here, and like this way separation will takes place. Separation will takes place, and here these two are moving in upward direction, these two are moving downward like this way. Here one chromosome will be there, here one chromosome will be there, here one chromosome, here one chromosome. Then here centriole will be there, here is there, chromosome is there, this one is there, one cell, this one is there cell, this one is also cell, this one is also cell, this one is also cell. So here nucleolus, here nucleolus, here nucleolus, here also nucleolus, and this one is the chromosome, this one is there chromosome. You can say this one is there chromosome. So in this way we have studied. In the meiosis, first two daughter cells were produced, and these two daughter cells were having two chromosomes, and these two chromosomes after metaphase second, their chromosomes are brought to the equatorial plane. In the anaphase second, these chromosomes split, and sister chromatids are moving upward, downward, and from that, in the what you can see, telophase, this one is there, telophase second. In the telophase second, 4 haploid cells are produced. How many cells are produced? 4 haploid cells are produced, and these haploid cells you are saying gametes. So in this we have studied in the prophase first, prophase second, prophase third, in the prophase and prophase first in case of the meiosis, and meiosis first and meiosis second. So in the meiosis first, which stages we have studied? Meiosis first in the prophase first. Prophase first is there having 5 stages: liptotin, zygotin, dikinesis and diplotin. diplotin and dikinesis. These 5 stages we have studied, and after that metaphase first, then anaphase first and telophase first, and after that we have studied meiosis second. In that prophase second, metaphase second, anaphase second, and the telophase second. Like this way, in the meiosis, from one cell, 4 different types of cells are produced, and these are haploid, and this is called as a meiosis. So in case of the meiosis, if you will watch this video again and again, you will understand, and if you will not understand, you can ask, I will explain this one again. So points we are going to write everything.